Hi, I'm Karen Situ, a fourth-year undergraduate student at UC Davis. Today I'll be discussing invasive grasses and the impact they have on native species in response to both competition and drought, as well as the role root traits play in that. So before I start, I want to preface this by saying that this experiment was designed by my mentor, Justin Luong, who allowed me to analyze some of the data from his project. This experiment was a greenhouse experiment where native and invasive plants from grasslands were grown together in a pot. In high competition scenarios, there were two of each native plant and 10 invasive plants, while in low competition scenarios, there were two of each native plant and two invasive plants. After some time, half of the pots were subject to drought conditions in which they received no water. The plants would then be harvested from each pot and various traits were measured, like root traits, which is my main focus for this project. The measurements that I primarily worked on were scanning roots on a program called WinRhizo, which would give us information about the root like root length, volume, and so on. Since root traits aren't as well studied, I wanted to see if they were significant in determining plant productivity and to see if functional root traits are a viable measure that can be used to later on determine strategies for invasive plant management. Afterwards, the data that were collected was analyzed with Elmer tests and visualized with interaction plots. We found that two of the significant traits were below-ground biomass and root tissue density in analysis of all plants. The first example I'm showing right now of a significant root trait is below-ground biomass under an interaction effect between treatment and competition. Below-ground biomass is important to study because it can tell us how much investment the plant is putting into root production. In the graphs comparing the below-ground biomass of plants grown with HOLA and BRMA, we can see that while grown in low competition, both invasives had a similar pattern of increase in below-ground biomass when well watered. When in high competition, plants grown with HOLA also had the same pattern. However, BRMA shows the opposite pattern where there is less below-ground biomass under high competition and well watered. The second root trait I'm going over is root tissue density under an interaction effect between treatment and competition. We can see that in a general analysis of all species, there are differences in response to competition and drought on root tissue density, as well as differences in response to each respective invasive grass. High competition and drought had a tendency to reduce root tissue density. For plants in the holopods, they tended to increase in root tissue density under well-watered conditions and increase in root tissue density under low competition. Plants grown with BRMA behave similarly in low competition environments, but had little change in high competition environments. Overall, HOLA had higher below-ground biomass when well-watered, meaning that it had more resources to allocate to its roots, while BRMA roots under high competition had less below-ground biomass when well-watered, indicating that they may have dedicated less resources into its roots since water was plentiful and it didn't need to compete with other plants as much for water. HOLA plants and BRMA plants both had increased root tissue density when under low competition and well-watered conditions, but while HOLA plants followed the same pattern under low competition, BRMA stayed relatively the same no matter the watering condition. This may indicate that under high competition, all the BRMA plants prioritize nutrient uptake, which differs from HOLA plants. Since we can see that the responses of HOLA and BRMA are so different, it shows that the approach that we will want to take in managing invasive plants could be different according to the species. In the example highlighted with below-ground biomass, BRMA had lower below-ground biomass under well-watered and high competition conditions, while HOLA had higher below-ground biomass in well-watered and high competition scenarios. So depending on how the climate changes, one species may need to be prioritized more than the other for removal. Also shows that we can draw conclusions that we wouldn't have been able to before about plant behavior and response from root traits, making it a potential tool to help determine what actions to deal to invasive species, such as determining if we should prioritize one species or even if we should consider removing it altogether. Lastly, I want to give special acknowledgments to Justin Luong, my mentor and the person who conducted this experiment, and Jennifer Funk, the PI for this project at Funk Lab. Additionally, I wanted to thank USDA NIFA, who were the funders for this project. Thank you.